My name is Welcome to the Bearer of Bad News. I'm your host, Lore Bear, and I hate to be the bearer of bad news, so I won't. This is your weekly recap of positive news stories from around the world. Articles included in this broadcast were originally published from April 5th to 8th, 2026. The stories in this episode are sourced from various news outlets and publications. The Bearer of Bad News does not conduct independent fact-checking or verification of these stories. Listeners are encouraged to review original sources for accuracy and complete context. We are not responsible for the accuracy or content of third-party reporting, which is a fancy way of saying, I read it, I didn't report it, and here are this week's stories. First up, we have these common drug tests lead to tens of thousands of wrongful arrests a year, experts say. One state is fighting back. From edition.cnn.com, bird poop scraped off a man's car appeared on a drug test as cocaine. A toddler's ashes registered as methamphetamine or ecstasy. And a great-grandmother's medicine tested positive for cocaine, spawning a 15-month legal nightmare, forcing her to refinance her home and spurring a new state law that could set a precedent across the country. Colorado just enacted the nation's first law banning arrests based solely on the results of colorimetric drug tests, a field test widely used by law enforcement across the country. The tests are popular because they're cheap, portable, and can screen for drugs in mere minutes. It's not just feasible to send all suspected drug samples to state laboratories, which would be far more expensive and could take days or weeks to return results. But these inexpensive tests also lead to false positives at alarming rates, researchers from the University of Pennsylvania found. While the actual error rate nationwide is unknown, previous studies by manufacturers have put it around 4%, but the UPenn researchers believe the actual rate is much higher, from 15 to 30%. 15 to 30? Okay. And a study by the New York City Department of Investigation showed test error rates from 79 to 91% in some correctional settings. So depending on the light. From lost jobs to months in jail, innocent people are at risk of having their lives derailed by these inaccurate tests, said Des Walsh, founder of the Roadside Drug Test Innocence Alliance. What are colorimetric drug tests? Colorimetric tests are approaches containing chemicals that change color when certain substances or compounds are detected. They typically cost between $2 and $10 and are used by law enforcement agencies across the U.S., Walsh said. By contrast, more reliable and portable electronic devices can cost from $24,000 to $80,000, hence the continued reliance on a $2 roadside drug test, Walsh said. While the design can vary by manufacturer, the pouches contain different chemicals that are intended to react to the compounds found in different narcotics, Walsh said. So there are tests for cocaine, for methamphetamines, for heroin, for fentanyl. But many everyday items have compounds that can also trigger a positive result. For example, the chemical characteristic of amphetamine has similar characteristics to the chemical compound for sugar, Walsh said. That's why sugar and sugar substitutes can show up as controlled substances on colorimetric tests. Even colorimetric test makers say their products only screen for the possibility of illegal drugs. They should not be considered tools for verification. Note, all test results must be confirmed by an approved analytical laboratory. Reads one warning label for a pack of colorimetric tests. The results of this test are merely presumptive, it says. Reactions may occur with, and such compounds can be found in, both legal and illegal products. Still, horror stories have emerged when ordinary people are arrested and jailed over colorimetric test results. Bird droppings and human remains test positive for drugs. Former Georgia Southern University quarterback Shai Wertz was suspended from the team after he was pulled over for speeding in South Carolina and charged with cocaine possession, CNN affiliate WXIA reported. During the traffic stop, a deputy noticed white residue on the hood of Wertz's car and ran it through a colorimetric test. The sample tested positive for cocaine. Wertz fought the case, and a lab tester later confirmed the substance he said was bird poop was not cocaine. His suspension was lifted, and the deputy resigned months later. Another traffic stop led to officers claiming a 2-year-old girl's cremated remains were methamphetamine or ecstasy. Illinois officers pulled over Dartavius Barnes and found a small urn in his vehicle. I checked for cocaine, but it looks like it's probably Molly. A slang for ecstasy, one officer said on body cam video. When police showed Barnes the urn, he pleaded with them to give it back. No, 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 bro. That's my daughter, he said. Okay. Poor lighting, especially at night, can make results of colorimetric tests difficult to interpret, Walsh said. In Florida, Kenaz Edwards was jailed because he couldn't pay the $178,000 bond set after a bottle of lidocaine found in his car tested positive for cocaine, the Florida Times-Union reported. He spent more than 3 months behind bars before the local district attorney office only tested samples in a lab after a trial date was set. But colorimetric drug tests aren't limited to traffic stops. Studies show false positive rates are especially high in prisons, which can lead to unfair punishments for inmates. Many samples taken from incoming mail have contaminants on the paper or in the ink, Walsh said. Commissary items such as soap, chocolate, and coffee can also test positive. Even children can be victims of false positive tests. In Iowa, a 13-year-old girl was expelled from school last year because the cookies she brought to school tested positive for THC on a field drug test, Walsh said. Who's testing 13-year-old girls' cookies for pot? I'm sorry. What? The cookies were tested because another student felt sick, though the cause of that illness was unclear. Regardless, the 13-year-old couldn't go back to school for months. It took her mom months to get a laboratory test that proved there was no THC or any illegal substance in the cookies at all, Walsh said. And in Colorado, a retiree's nightmare became a catalyst for change. A hospitalized great-grandmother was charged with cocaine possession. Holly Bennett was already in severe distress when a police officer entered her room and accused her of stashing cocaine in her purse. She suffered from a bone infection from my cervical spine all the way down to my hips and pelvis, the 65-year-old Colorado said. Bennett underwent emergency surgery on her neck because my arms were getting paralyzed. As she slowly woke up from surgery, Bennett was startled to see an officer in her room. He said, "I'm with the Lafayette Police Department, and I'm here to issue you a ticket for possession of cocaine," Bennett said. When Bennett insisted she didn't have cocaine, he said, "Well, that's what you had in your purse," she recalled. It's not clear who inspected Bennett's purse and reported the contents to authorities. Due to federal privacy laws that protect sensitive patient information, we are unable to respond to specific patient questions, Intermountain Health said in an email to CNN. Circumstances can arise where conducting a personal belongings search is needed to maintain a safe and secure care environment for all patients, caregivers, and visitors, the hospital network added. Lafayette Police Sergeant Jeremy Molander said he did not find records for Bennett's October 2022 case. After a 2020 state law reduced penalties for certain drug offenses, he said officers in such cases typically issue a summons instead of making an arrest. But Bennett said she shouldn't have to fight the legal system at all. What I had in my purse was a few Ritalin that were prescribed to me. It's in my prescription records, and they had broke down just from movement from me throwing my little bag around. Investigators could have checked her blood test at the hospital, which detected only her regular medication and not cocaine, she said. The next 15 months were pretty miserable as the disabled senior fought her case and begged to have the sample tested in a lab. She refinanced her home to free up money as she struggled to find an affordable attorney. I kept going to court with a lawyer, Bennett said. There, prosecutors pressured her to plead guilty in exchange for diversion, a 6-month program that, if completed, would erase the drug charge from her record. She said, but diversion programs can cost innocent people time and money through no fault of their own. For those who get arrested due to colorimetric testing, over 90% of people are taking a plea deal because they can't afford to remain in jail and wait 6 months for laboratory tests, Walsh said. Bennett even offered to pay $500 for a lab test, even though she really didn't have it. It would have been something that would have hurt our ability to pay utilities, she said. At the time, the state lab was so busy that samples were only tested if cases were set for trial, Boulder County District Attorney Michael Doherty said, so that was part of the delay as well. After several months, Bennett found an attorney, her trial date was set, and the substance from her purse was examined in a lab. In the end, she didn't have to pay to retest the sample, I guess because there was no cocaine in it. The district attorney's office dismissed Bennett's charge, Doherty said, and records relating to her case were expunged per Colorado law, including the original police report. They were wrong and I was right, Bennett said. But she worries about others who could be wrongfully charged, losing their jobs, and languishing in jail for months while waiting for a trial date or for a lab test. The district attorney agreed and said the system was flawed, so his office teamed up with Bennett's attorney, lawmakers, and the Corey Wise Innocence Project and law enforcement officials to find a solution. Doherty said he had no idea how inaccurate colorimetric tests could be until Bennett's case, which came a year before the UPenn study was released. With what we know now about the colorimetric field drug test, we're going to be much more careful about whether the results that indicate it's positive are accurate or not, the district attorney said. Most law enforcement agencies in Boulder County don't use colorimetric tests, he said, but others in Colorado do. The top deputy in his office joined a state task force that helped craft legislation banning officers from arresting someone based solely on results from those presumptive field tests. Everyone at every stage seemed to blindly trust the results of this test, said Bennett's attorney Noah Stout, who also served on the state task force. Stout said lawmakers were already considering possible new legislation before learning about Bennett's case, but the great-grandmother's ordeal kicked it into special gear. It's sad that it takes an old lady in a walker who has no drugs in her system to demonstrate that this is wrong, Stout said. There are also a lot of people who don't check all those sympathetic boxes, who are more likely to face consequences and don't have the means to retain private counsel or have the will to fight the system. There's a huge gap between the individuals who qualify for a public defender versus who can actually afford an attorney. Governor Jared Polis signed the bill into law last week. The statute is the first in the nation to address colorimetric drug testing, the National Conference of State Legislators told CNN. Up next, research turns recovered car battery acid and plastic waste into clean hydrogen. From cam.ac.uk, researchers have developed a solar-powered reactor to break down hard-to-recycle forms of plastic waste, such as drink bottles, nylon textiles, and polyurethane foams, using acid recovered from old car batteries and converting it into clean hydrogen fuel and valuable industrial chemicals. The reactor, developed by researchers from the University of Cambridge, is powered by the energy of the sun and could be a cheaper, more sustainable alternative to current chemical-based recycling methods. The team say their method could create a circular system where one waste stream solves another. The results are reported in the journal Joule. Global plastic pollution exceeds 400 million tons per year, and yet only 18% is recycled. What? Who's not recycling? All right, I want your names. Put them in the chat. The rest is burned, landfilled, or leaks into ecosystems. The researchers say that their method, known as solar-powered acid photoreforming, could help address the global mountain of plastic waste. The researchers engineered a photocatalyst that is robust enough to withstand the highly corrosive effects of acid while making productive use of the acid inside spent car batteries, which is normally neutralized and discarded. The discovery was almost accidental, said Professor Erwin Reisner from Cambridge's Yusuf Hamid Department of Chemistry, which led the research. We used to think acid was completely off limits in these solar-powered systems because it would simply dissolve everything. But our catalyst developed didn't. And suddenly a whole new world of reactions opened up. Acids have long been used to break plastics apart, but we've never had a cheap and scalable photocatalyst that could withstand them, said lead author Kay Kwarting, a PhD candidate in Reisner's research group who developed the photocatalyst. Once we solved that problem, the advantages of this type of system became obvious. The method developed by Kwarting, Reisner, and their colleagues first treats waste plastics with the car battery waste acid, breaking the long polymer chains into chemical building blocks such as ethylene glycol, which the photocatalyst then converts into hydrogen and acetic acid, the main ingredient in vinegar, when exposed to sunlight. In laboratory tests, the reactor generated high hydrogen yields and produced acetic acid with high selectivity. It also ran for more than 260 hours without any loss in performance. The approach works for multiple types of plastic waste, even those that are currently tough to recycle, such as nylon and polyurethane. This offers a real advancement to current upcycling technologies that do not cover plastics beyond PET. Also, a system that does any sort of melting or whatever, it doesn't need to break down the plastic, doesn't need to chop it into smaller bits, is going to be an advantage because we can't do thin plastics that way. But this kind of system, you should be able to just scrunch up the thin plastic into a ball and throw it in. Perfect. The approach works not just with new laboratory-grade acid, but with the acid recovered from car batteries. These batteries contain between 20 to 40% acid by volume and are replaced worldwide in huge numbers every year. The lead in these batteries is typically extracted for resale, but the acid creates extra waste once it is safely neutralized. It is an untapped resource, says Kwarting. If we can collect the acid before it's neutralized, we can use it again and again to break down plastics. It's a real win-win, avoiding the environmental cost of neutralizing the acid while putting it to work generating clean hydrogen. The researchers say their method offers a potential order of magnitude cost reduction compared with other photoreforming approaches, largely because the acid enables increased hydrogen production rates and can be reused rather than consumed or wasted. Kwarting says that although challenges remain, such as ensuring reactors can withstand corrosive conditions, the fundamental chemistry is sound. These acids are already handled safely in industry, he said. The question now is engineering. How do we build reactors that can run continuously and handle real-world waste? The researchers say that their approach won't replace conventional recycling, but it could complement it by handling contaminated or mixed plastics that currently have no viable route to reuse. We're not promising to fix global plastics problem, said Reisner, but this shows how waste can become a resource. The fact that we can create value from plastic waste using sunlight and discarded battery acid makes this a really promising process. Up next, 10% of the ocean is protected, now just 20% more to go. From news.mangabay.com, more than 10% of the world's ocean is now protected to some degree, marking significant progress in global ocean conservation efforts, according to an April 1st announcement from the UN Environment Programme's World Conservation Monitoring Center, UNEP-WCMC. However, this accomplishment comes 6 years behind schedule, and experts warn that efforts must pick up speed to meet the more ambitious current goal of protecting 30% of the ocean by 2030. Achieving that will require protecting an additional area about the size of the Indian Ocean within the next 4 years. The crossing of the 10% threshold follows the recent addition of 284 marine and coastal protected areas in Indonesia and Thailand to the World Database on Protected and Conserved Areas, WDPCA, a platform managed by the UNEP WCMC. This database tracks progress towards Target 3 of the Kunming Montreal Global Biodiversity Framework, also known as the 30 by 30 goal, which aims to protect nearly a third of the ocean as well as Earth's land and inland waters by the end of the decade. Prior to the creation of the 30 by 30 goal, governments were focusing on Aichi Target 11, which called for protecting 10% of land and water by 2020. The tracking system itself has recently changed. Formerly known as the World Database on Protected Areas, the WDPCA was updated in November to mark its merger with the World Database on Other Effective Areas-Based Conservation Measures, WDOECM. The merger effectively combined formal protected areas with the OECMs, areas not necessarily protected for conservation purposes, but that may still deliver benefits for the environment. Some experts argue that many OECMs fall short in terms of suitability and consistency with conservation standards, which has generated debate about counting them as protected areas. Currently, OECMs only account for about 0.22% of total ocean protection, according to UNEP WCMC. One of the newly reported marine protected areas, MPAs, that helped push the world over the 10% mark, according to WDPCA, is the Timur, Patani, Palau, Saifi Patani MPA, known as the Patani Bacoli, and Saifi Island MPA. That protects 3,372 kilometers, 1,303 kilometers square, mile square, of sea and coastal area in North Maluku province of Indonesia. Other recent additions to WDPCA that contributed to the 10% milestone include the 4.5 million kilometers squared, 1.7 million miles squared Tainui Atea MPA, which covers nearly the entirety of French Polynesia's exclusive economic zone and is currently the largest MPA in the world. Australia has also made a sizable contribution with the recent reporting of several new MPAs. These include a 400% expansion of its Heard Island and McDonald Islands Commonwealth Marine Reserve in subantarctic waters to cover 380,654 kilometers squared, or 140,971 miles squared, an area twice the size of the U.S. state of Florida. Both French Polynesia and Australia are among 33 countries and territories that have protected more than 30% of their waters, according to WDPCA data. Go Australia and Polynesia, French Polynesia! We all depend on the ocean for our survival, Neville Ash, director of UNEPWCMC, said in a statement. The great strides at the national level over the past 2 years to protect more than 10% of the marine realm is therefore a moment for celebration, but reaching this milestone is a reminder of how much work there is still to do. Of course, Australia is an island nation. Let's not forget that, people. While the global ocean has now surpassed 10% protection, data is lacking on how effective these protected areas are, according to UNEP-WCMC. Target 3 doesn't just call for 30% of the oceans to be within PCAs, Ash told Mongabay in an email referring to protected areas or conserved areas. It calls for 30% of the ocean to be within effectively conserved and managed and equitably governed PCAs. Although we have good data on the extent of PCAs, data on how effective they are is much more limited. It's vital that the aspects of these targets receive equal attention to coverage over the next coming years. Another global marine protection database, the MP Atlas, which tracks progress in marine protected areas using WDPCA data, indicates that as of March 2026, only about 3% of the ocean is fully or highly protected, meaning that extraction or other potentially destructive activities are entirely prohibited from these areas. However, Lance Morgan, president of the Marine Conservation Institute and the U.S. based group behind the MP Atlas, says the U.S.'s recent decisions to allow commercial fishing in its four large Pacific Marine national monuments, which had been previously highly protected, might reduce the fully and highly protected figure by 0.5 to 0.7%. That is, if the decisions survive legal challenges. In all other protected or conserved areas, activities such as fishing are often still permitted. There's a lot of different levels of protection and different labels that people can apply to ocean protection. Dan Crockett, executive director of the UK-based Blue Marine Foundation that campaigns for 30% marine protection by 2030, told Manga Bay. The same applies for ECMs, other effective conservation measures. They can both be a good or a bad thing. It all depends on what they do and the sort of activities that are permitted within them, and whether they are generally working to protect marine life. Crockett said Blue Marine prefers to work with partners looking to create highly or fully protected areas because these offer the swiftest way to protect biodiversity and meet many of the goals of conservation. Morgan said that he was pleased to see the number of marine protected areas growing, but that more progress has to be made. If 2030 is really the deadline, we are extremely far behind, Morgan told Manga Bay, and we really need to up our efforts. So far, marine protected areas are overwhelmingly concentrated within national waters, accounting for more than 9% of the global ocean, while the high seas remain sparsely protected, accounting for only about 1% of the total. Up next, Johns Hopkins scientists developed nasal DNA vaccine for tuberculosis. From SciTechDaily.com, a new experimental vaccine takes aim at one of tuberculosis's most stubborn defenses, the ability of bacteria to persist despite treatment. In a study published in the Journal of Clinical Investigation, researchers from Johns Hopkins Medicine and the Johns Hopkins Bloomberg School of Public Health reported developing a therapeutic DNA vaccine delivered through the nose for tuberculosis. The vaccine combines two genes designed to train the immune system to target drug-tolerant bacterial persisters, which can survive long courses of antibiotics and lead to disease relapse. TB has affected humans for at least 6,000 years. The World Health Organization estimates that about 1/4 of the global population, roughly 2 billion people, carry the latent infection without symptoms. In 2024, more than 10 million people developed active TB and 1.2 million died from the disease, making it the leading cause of death from a single infectious infection. The need for better treatment strategies. WHO has emphasized the need for therapeutic vaccines that can be used alongside existing drug treatments to shorten therapy and improve outcomes. Current TB treatment often requires long multi-drug regimens that are difficult to complete, and drug-resistant strains continue to spread. The new vaccine from Johns Hopkins may help address these challenges. Administered together with the first-line TB drug therapy, our intranasal DNA fusion vaccine helped infected mice clear the disease bacteria faster, reduced lung inflammation, and prevented relapse after treatment ended, says study lead author Styliani Karankia, MD. An assistant professor of medicine at the Johns Hopkins University School of Medicine. The vaccine also helped the powerful TB drug combination of bedaquiline, protomidine, and linezolid work better, suggesting it could be used with treatments against drug-resistant TB to help the body fight the disease, even hard-to-treat cases. How the vaccine works. According to Karanika, the vaccine merges two genes, REL, MTB, and MIP3A, and is administered through the nose to take advantage of several biological effects. First, TB bacteria possess a gene, REL-MTB, that produces a protein, REL-MTB, but not in italics, to help the microbes survive hostile conditions such as an antibiotic exposure, low oxygen, and nutrient limitation by entering a drug-tolerant persistent state, she says. Fusing REL-MTB with the MIPA3 gene produces a signal that attracts immature dendritic cells, key cells that pick up TB proteins and present them to T cells, the immune cells that help coordinate a targeted attack on the TB bacteria. Finally, intranasal delivery focuses vaccination on the respiratory mucosa in the lungs, where TB infection occurs, helping generate long-lasting, localized T-cell immunity in the airways and lungs, along with systemic immune responses, says Karanika. By combining these mechanisms, the researchers aim to boost immunity activity directly in the respiratory tract. In mouse studies, the vaccine increased the recruitment and activation of dendritic cells and improved how these cells interacted with T cells in the lungs. It also produced strong, long-lasting immune responses both in the lungs and throughout the body, involving CD4 helper T cells and CD8 killer T cells. Findings from primate studies. In rhesus macaques, the intranasal DNA vaccine triggered measurable TB-specific immune responses in both the bloodstream and the airways. These responses were similar to those associated with reduced bacterial levels in mice. The immune activity lasted at least 6 months, suggesting the vaccine's effects may be durable. However, Karenica notes that these studies measured immune responses only and did not test protection against actual TB infection. She says additional research is needed before the vaccine can move into human clinical trials. These non-human primate data are encouraging because they show that the MIP3A/RELMTB vaccine can generate durable antigen-simulated immune responses in an animal model whose immune system more closely resembles that of humans, says Karenica. That gives us an important transitional bridge between the mouse efficacy studies and the additional preclinical work needed before human trials. The researchers say their results support a broader approach that targets TB persisters with immunotherapy instead of relying on only antibiotics to kill actively growing bacteria. DNA vaccines are relatively stable and can be produced effectively, which could make them practical if this strategy proves successful in humans. Up next, from biodiversity to carbon capture, saving bison from extinction has brought big benefits to Europe. The European bison is making a comeback across the continent, bringing with it a wealth of wildlife benefits. Once widespread, Europe's largest wild land mammal was driven to the brink of extinction in the early 20th century by hunting and habitat loss. Now herds can be found in the UK, Romania, Germany, Switzerland, Poland, Belarus, and Lithuania. The resurgence of the European bison. The European bison, or wisent, once inhabited much of the European continent before populations were decimated by hunting. When the last wild European bison was shot in the Caucasus in 1927, there were fewer than 60 individuals alive in zoos and private parks, according to Rewilding Europe, an organization dedicated to creating rewilded landscapes. European bison began to be reintroduced back into the wild as early as the 1950s and has become one of Europe's most successful wildlife recovery stories. Over the last 10 years, the estimated number of free-roaming European bison has increased from 2,579 to 7,000 individuals, with the largest herds found in Belarus and Poland. Rewilding Europe efforts have resulted in over 100 European bison now roaming free across the southern Carpathians of Romania, while Rodthorpe Mountains of Bulgaria have been home to a small but growing wild bison population since 2019, the first time the animal has roamed free there since the Middle Ages. Bison boost biodiversity in UK woodland. The reintroduction of bison into Europe is also having a wider positive impact on the environment. In the UK, a herd was rewilded in the Blean Woods, Kent, in 2022. Just a few years on, they are already having a great impact, conservationist Hannah Mackins told UK newspaper The Independent. The animals aid woodland regeneration and manage habitats for other wildlife by grazing, felling trees, stripping bark, and taking dust baths. This boosts biodiversity and restores complex ecosystems, rendering environments more robust in the face of climate change. Mackin says the effect is really noticeable. Walking through there, we have a lot more light on the woodland floor. We have species growing through that wouldn't have had before because it was so dense with trees, bracken, and brambles, she said. Bison bridges, 2 of which are already installed and 2 of which will be constructed by the end of the year, hope to allow the herd access to a much wider area of the woodland. Romania's bison help capture carbon. After disappearing from Romania more than 200 years ago, rewilding Europe and World Wildlife Federation Romania reintroduced bison to the Carpathian Mountains in 2014. A 2024 study from Yale University suggests these massive herbivores could have a role to play in mitigating the impact of climate change. The herd of 170,000 individuals could potentially be helping capture and store the carbon released by up to 84,000 average U.S. petrol cars each day. I like that they specify U.S. petrol cars. They do this through a combination of evenly grazing grasslands, recycling nutrients which fertilize the soil, dispersing seeds, and compacting the soil to prevent carbon from being released. Researchers say that, having evolved alongside this ecosystem for millions of years, their removal upset the delicate balance, causing carbon to be released. Really? The report's authors noted, however, that their estimated figure could be up to 55% higher or lower. The higher figure is equivalent of around 84,000 US petrol cars annually, and the median average is 43,000 cars. I don't think we needed to be specified about that. Bison in the Netherlands bring benefits to songbirds. In the Netherlands, bison are lending a surprising hand to songbirds during breeding season. In the South Kennemerland National Park, passerine birds upcycle the winter coats that the herd sheds in spring to construct their nests. According to the Wisdom Project Kransflak, which manages the area, bison hair is favored because it is woolly and soft, providing good insulation and maintaining the temperature for the eggs. It says applied biology students are now researching whether this nesting material has an effect on the breeding success of the birds. Up next, Ecuador study finds tropical rainforest biodiversity rebounds over 90% in 30 years. Tropical rainforests, from phys.org, tropical rainforests are home to almost 2/3 of all vertebrate species and 3/4 of all tree species. They are the most species-rich terrestrial ecosystem on Earth. However, over half of these diverse rainforests have already been cleared, and their area continues to decline drastically, primarily for agricultural purposes. Is there a chance of regeneration? And can not only trees, but also the unique diversity of thousands of animal species return to cleared areas? The answer is surprisingly clear-cut and encouragingly positive. Trees regrow rapidly on agricultural land. As soon as land use ceases, a diverse range of animal species also reestablish themselves. Biodiversity recovered more than 90% of its original level within 30 years. During this period, as many as 3/4 of animal and plant species typical of primary forests return. The paper is published in the journal Nature. Teams led by Professors Thomas Smit and Jorg Mueller from the University of Würzburg Biocenter contributed to the publication. Study conducted in northwestern Ecuador. The group's findings apply, at least, to the study area in Chaco in northern Ecuador. In this region, there are still a few patches of undisturbed primary forest and larger areas of secondary forest. These form the reservoir for many of returning animals and plant species. Researchers from more than 30 universities and institutions have, for the first time, conducted a detailed study of the natural regeneration of 16 different groups of organisms, animal, plant, and bacterial species along a regeneration gradient. A total of 62 sites were compared, all of which have been protected for several decades by the conservation organization Joko Toko and integrated into a large nature reserve, actively used pastures and cocoa plantations, secondary forests of varying ages that were formerly used as pastures and plantations, and untouched primary forests. Rainforest resilience proven empirically for the first time. Lead author Timo Metz carried out and summarized the extensive analysis as part of his PhD at Darmstadt Technical University. He emphasizes rainforests as complex ecosystems and species-rich communities demonstrate remarkable resilience and the ability to return to their original state. This stability has often been modeled theoretically, but until now could not be demonstrated on the basis of such extensive empirical data. Senior author Nico Bluthgen is professor of ecology at TU Darmstadt, spokesperson for the Reassembly Research Group, and alumnus of the University of Würzburg. He initiated the studies together with Dr. Martin Schaefer, director of the Ecuadorian conservation organization Jokoto. Nico Bluthgen states, the many rapidly returning animal species are not only beneficiaries of forest regeneration, but are also its key agents. Bats, monkeys, and other mammals, as well as birds, bring tree seeds back to the cleared areas. Dung beetles bury the seeds in the soil, and hundreds of other animal species ensure pollination. Martin Schaefer adds, our findings that 75% of species composition and 90% of species diversity return under their own steam with a single human generation show just how effectively we can protect nature. By purchasing and protecting land, we can preserve the diversity of life and the foundations of our societies, soil, water, and the pollination of the plants that form the basis of our food supply. Up next, inside a Kentucky city's unusual experiment in citizen-led governance. Public trust in government is near historic lows, but Americans' trust in their local government far outweighs trust in the federal government. It's been this way since the mid 2000s when the State of the Nation project began keeping track. Standing on the sidelines of Kentucky's first civic assembly and one of the first carried out anywhere in the country, the palpable excitement I saw in that conference room was a far cry from the profound polarization, distrust, skepticism, and fatigue driving American political disengagement. For the past month, 36 randomly selected residents of Lexington have been meeting regularly at Transylvania University to develop and deliberate over policy recommendations for revisiting Lexington's charter to produce healthier and more effective local governance. There's a lot of attention at the federal level, but there's also a sense of a lack of control. Marjan Asasi, executive director of the Federation for Innovation in Democracy, FIDE, in North America. She says local co-governance models like these are part of the solution to rebuilding American democracy from the ground up. People are feeling like I can control my local government, I can control what's happening locally. The assembly was coordinated by Civic Lex, a local nonprofit working to boost civic health and engagement in Kentucky's second largest city. Full disclosure, I am reporting fellow with Civic Lex, but I was not involved in developing or coordinating the assembly. Across 7 sessions, the Assembly members learned about the people and systems that keep Lexington's roads paved and City Hall lights on. They heard from subject matter experts and members of the public. They built mutual trust, changed their minds, and came to difficult compromises. And when the Assembly issued its final recommendations on March 29th, members ultimately decided to trust their council representatives enough to recommend a large pay bump in addition to pushing for increased attendance and accountability requirements. The slow rise of civic assemblies in America. Civic assemblies aren't anything new. Athenian democracy has guided participatory government for centuries. Modern versions of these deliberative forms have been used around the world for the past 3 decades, especially in Europe. They've been slower to catch in the United States, though a handful of local initiatives have been convened, such as an assembly on youth homelessness in Oregon or one on childcare in Colorado. FIDE North America is one among a number of organizations, including Colorado State University's Center for Public Deliberation and the Hannah Arendt Center for Politics and Humanities, that is working to facilitate the movement spread there. Asasi says that across North America, proving the efficacy of co-governance to local governments has been an uphill battle. When I first started with FIDE, it was hard to sell assemblies. It was hard to make the case for why traditional ways of engagement weren't sufficient, says Asasi, whose organization works with cities, including Lexington, to conduct independent assemblies reviews to ensure that processes are equitable and the final recommendations would receive a fair shot in front of council and voters. Now I feel like most are acknowledging that there was a recognition that the traditional ways of engaging with constituents is just not working anymore. 2024, when city council members in Fort Collins, Colorado, needed to decide on how to use the land beneath a demolished football stadium, they voted to convene a civic assembly. Assembly members' recommendations developed a multi-use site featuring a bike park, a wildlife rehabilitation center, outdoor community spaces, and indigenous cultural representation successfully passed last year. People are hungry for new ways to engage, and I think it's really clear that the status quo is not working, says Holly Royson-Gilman, a senior fellow at New America who researches civic engagement and co-governance. I'm not about replacing representative democracy, but it's about augmenting it and creating richer, more additive civic experiences where people feel seen and heard in their day-to-day lives. Up next, man unexpectedly cured of HIV after stem cell transplant. TechFixated.com. A 60-year-old man from Berlin is now HIV-free. He has been off all medication for more than 7 years. His immune system shows no signs of the virus. His antibody response to HIV is fading, which doctors say is exactly what happens when there is nothing left for the body to defend against. Published in the journal Nature on December 1st, 2025, this case has been confirmed as the 7th documented cure of HIV in history. But what makes it generally remarkable is it's not simply that he is cured, it is how he was cured, because the stem cells used to treat him were not supposed to be able to do this. A discovery that rewrote the rules. For nearly 2 decades, the scientific understanding of how HIV could be cured through stem cells transplantation resisted a single protein. That protein is called CCR5. HIV, to infect immune cells, needs to attach to a receptor on the cell's surface. For the most common strains of HIV, CCR5 is that receptor. It is the door the virus walks through. In the early 1990s, scientists discovered that roughly 1 to 2% of people in Northern European descent carry a mutation in both copies of the CCR5 gene, a variant called CCR5 delta 32. This mutation effectively removes the receptor from the cell surface entirely, making those individuals almost completely immune to the most common forms of HIV. The first person ever cured of HIV, Timothy Ray Brown, known as the Berlin patient, was treated in 2008 at the Charité Hospital in Berlin. His doctors had what seemed like an inspired idea. Brown had both HIV and acute myeloid leukemia. He needed a stem cell transplant to treat the cancer. What if the donor had the CR5 delta 32 mutation? The transplant would replace his immune system with one that HIV could not infect. Brown and a handful of others received special donor stem cells carrying this mutation in both copies of the CCR5 gene. To many scientists, these cases suggested that CCR5 was the best target for an HIV cure. 5 cures followed using the same approach, all relying on donors with both copies of the CCR5 mutation. Scientific consensus hardened. You need those cells to cure HIV this way. Without them, the transplanted immune system would simply become infected by whatever residual virus remained in the patient's body. The logic was airtight until it wasn't. How this patient's case was different. The man at the center of the December 2025 paper was diagnosed with HIV in 2009. In 2015, he developed acute myeloid leukemia, a blood cancer requiring urgent treatment. He needed a stem cell transplant. His doctors researched for a donor who carried both copies of the CCR5 delta 32 mutation. They could not find one. His doctors could not find a matching stem cell donor who had mutations in both copies of the CCR5 gene, but they found a female donor who had one mutated copy, similar to the patient. That matters because CCR5 delta 32 behaves recessively. Carrying one copy does not eliminate CCR5 from the cell surface. The donor cells still express the CCR5 receptor that HIV uses to gain entry. By every previous understanding, that should have meant the virus would eventually find its way back. The man received the transplant in October 2015. His leukemia responded well. In 2018, 3 years after the procedure, he made a decision that surprised his medical team. He chose to stop taking his antiretroviral drugs. He felt that he had waited some time after the stem cell transplant. He was in remission for the cancer, and he was always feeling that the transplant would work. Shortly after, the team found no signs of the virus in blood samples from the man, and the virus did not come back. It has now been more than 7 years since he stopped antiretroviral therapy. No evidence of HIV-1 replicating in the patient was found 6 years after the transplant. He has the second longest HIV-free period of any of the 7 cured individuals, behind the only original Berlin patient who has been free of the virus for approximately 12 years. What happened inside his body? The central question the December 2025 paper grapples with is how did a man whose new immune cells were not protected from HIV end up cured of HIV? The leading hypothesis involves a process called graft versus host effect, an immune reaction that is usually considered a dangerous side effect of stem cell transplantation, but which may, in this case, have played a crucial role in eliminating the virus. Researchers found that he had natural killer cells from his innate immune system with an unusual genetic profile, which prompted the development of a set of antibodies that were exceptionally efficient in seeking out and destroying whatever HIV-infected cells were left in his body after the stem cell transplant. In other words, the new immune system recognized the patient's remaining original immune cells as foreign and attacked them. Any cells still harboring HIV were destroyed in the process. The virus's reservoir, the hidden population of infected cells that makes HIV so difficult to eradicate permanently, was eliminated by not blocking the virus's entry into new cells, but by destroying the cells that had already carried it. These graft versus host reactions may have led to the viral reservoir being eliminated so effectively that the CCR5 delta 2 mutation is no longer necessary, as no virus capable of multiplying remains in the body. These hypotheses open up promising avenues for research aimed at achieving remission for HIV infection. How the study was conducted. The research team, led by Dr. Christian Gabler at the Charité Hospital in Berlin, conducted extensive virological and immunological analysis of the patient across the years following his transplant. They tested blood samples repeatedly for any signs of HIV DNA or RNA. They looked across virus-carrying cells capable of replication in both blood and intestinal tissue, one of the key reservoirs where HIV hides during suppressive treatment. They monitored the patient's HIV-specific antibody levels, tracking whether his immune system continued to mount a defense against the virus. Researchers have found no DNA in his cells capable of giving rise to new virus, and his antibody response to HIV is fading, indicating there is no virus for the immune system to respond to. The fading antibody response is a particularly significant marker. The immune system generates antibodies in response to an active threat. If HIV were still present somewhere in his body in any active form, the antibody response would be sustained. Its decline is strong evidence that the virus is genuinely gone, not simply suppressed. Along with our Wheel of Good Fortune edition is Let's Do the Good News Again. If articles are found during the making of the next set of episodes, we're going to add them to the list and report on them at the end of the next episode. Here are the stories we missed in last episode. First up, there's something special about Kangaroo Island koalas. From BBC.com on April 3rd, inside the island colony being tapped to help save Australia's koalas from deadly chlamydia. Brandishing long plastic poles, a small team of researchers are trying to coax a female koala down from the gum tree she's comfortably perched on. At first, she seems unfazed. Then everything happens quickly. She clambers down the trunk, leaps onto the grass, and lets out a deep growl before rolling onto her back, claws raised in defense. In a series of well-practiced steps, the experts carefully lift her into a crate. Once sedated, she's laid on a towel for a routine health check. I think she has chlamydia, says Karen Burke de Silva, a conservation biologist at Flinders University in South Australia. Chlamydia has become a major epidemic among koalas, affecting up to 88% of individuals in some mainland populations. Caused by the bacterium Chlamydia pecorum, it can lead to blindness, infertility, pneumonia, and unlike chlamydia in humans, which is rarely fatal, often death. Chlamydia has swept through the mainland, and this captured koala is one of about 40 inside South Australia's Bellary National Park near Adelaide, collared by scientists studying their health and genetics. Yet, on a nearby island, the disease has never been recorded. Kangaroo Island is thought to host the world's largest chlamydia-free koala population, offering something of a living insurance policy for the species. Still, these koalas are under a pressure of their own. More than a century of isolation has left them deeply inbred and genetically fragile. This is what brings Burke de Silva and her colleague Julian Beeman to study koalas in the region. They hope that by first improving the genetic diversity of Kangaroo Island's koalas, then introducing them to other low chlamydia areas of Australia, they can help tackle the current blight facing the species. Death by 1,000 cuts. Native to eastern and southern Australia, koalas are listed as vulnerable by the International Union for Conservation of Nature, though still numerous overall, with a population of between 398,000 and 569,000 according to official estimates. They have been declining steadily for decades and now mostly survive in small, fragmented populations. This has lowered their adaptability to the effects of climate change, habitat loss, and disease. Each of these pockets, you get inbreeding and random population fluctuations that raise the risk each one will go extinct, says Beeman. If we're not careful, it will be death by 1,000 cuts. Kangaroo Island is separated from Australia's southern coast by just 13 kilometers, 8 miles, and is the country's third largest island and a biodiversity hotspot. A short ferry ride from the mainland, the island's rugged coastline makes way inland for a patchwork of sheep-dotted grassland and dense native brush. The koala population living here is entirely descended from a small group of around 200 individuals introduced from the mainland in the 1920s at a time when conservationists feared that fur trade would wipe out the species. By 2019, the population had exploded to 50,000, so many that they were often described as a pest. People talk about them negatively, says Beeman, as we walk through a native eucalyptus forest on the island, but it was actually a highly successful introduction. As chlamydia runs rampant through mainland populations, the island's population has shielded its koalas from infection. Natasha Spite, a koala researcher from the University of Adelaide and the co-author of a 2019 study which found the Kangaroo Island population remains free of chlamydia, says this makes it the largest population in Australia with this status. While chlamydia may have existed in Australia before Europeans arrived in the late 18th century, research suggests that Westerners' livestock introduced new strains. Combined with habitat loss and shrinking genetic diversity, this has fueled an epidemic that threatens the species' survival. Chlamydia can be cured with antibiotics, but treatment is far from straightforward. It requires koalas to be captured, can fatally affect their ability to digest eucalyptus leaves, and provides no protection against reinfection. A vaccine approved in 2025 offers genuine hope, but cutting mortality in wild populations by 65%, but vaccinating wild populations at scale remains a formidable challenge. Researchers, including Spite, though, think that protecting Kangaroo Island's koalas by using them to help repopulate the mainland could, alongside the new vaccine, ultimately ensure the survival of the species. And as well, we have breeding knowledge, how indigenous expertise and Western science are converging, from theguardian.com on April 4th. I'm a glorified clam counter. So said Marco Hatch, a marine ecologist at Western Washington University and an enrolled member of the Samish Indian Nation. Hatch has been conducting surveys of mollusks growing in and around clam gardens in the Pacific Northwest as he collaborates with 7 indigenous communities to build or rebuild these rock-walled terraced beaches once created and tended by their ancestors. Hatch's surveys in service of this reclamation are rooted in Western scientific methodology and increased understanding about beach ecology and clam health. But just as important, the data Hatch provides can help these nations obtain the local, state, and federal permits they need to maintain or re-engineer these structures. And that helps them assert greater control over their heritage and regain food sovereignty for their communities. Rather than dismissing indigenous knowledge, more Western scientists are discovering its viability for themselves and adjusting their research goals to embrace it. That represents a massive shift, according to Kyle White, a professor of environmental justice at the University of Michigan and a member of the Citizen Potawatomi Nation. Historically, Western scientists have considered themselves rigorous and empirical, while they have classified traditional native thought as mythic, religious, or plain made up, he said. In fact, a long overdue braiding of native and Western knowledge is becoming ever more common. Prominent native authors such as Vine Deloria Jr. have pointed out native environmental practices in books for popular audiences. They've theorized, as the Alaskan native scholar Oscar Kawagli described it, native ways of knowing. More indigenous people, Robin Wall Kimmerer, author of Braiding Sweetgrass, is a notable example, are entering academia and changing it from the inside. While some tribal nations have hired their own scientists, non-native institutions are seeking to undo their erasure of indigenous cultures. The Brooklyn Botanic Garden has started to include labeling that highlights Lenape names and uses for food plants like persimmons. International environmental organizations also increasingly recognize the importance of including indigenous voices in discussions around the climate crisis. Since 2022, there's been federal funding to study ways to combine indigenous and Western sciences so each part remains distinct while being strengthened by the other. Keisha Supernant, who is Métis and Papachaise and the director of the University of Alberta's Institute of Prairie Indigenous Archaeology, said that Indigenous knowledge contained a rich history of observation, experimentation, and understanding that has its own systems of rigor. Such rigor is evident in places like the clam gardens that Hatch studies. Beginning at least 4,000 years ago, Native communities built clam gardens into the intertidal zone from Washington State University through coastal British Columbia and into southeast Alaska. They are a unique form of mariculture that provides harvestable habitat for an array of tasty ocean creatures like butter clams collected in great numbers, then smoked and dried and stored and traded. Hatch said, but they also yielded red rock crab, basket cockle, sea cucumbers, limpets, sea snails, and seaweeds in a veritable smorgasbord for humans and marine mammals such as otters. These gardens change where sediment moves and may protect against increasing shoreline erosion. Studies also show that clam productivity and populations are higher inside gardens than outside them. Although there have been instances where treaties affirmed their right to harvest, many indigenous groups lost access to the beaches where they once picked clams. Hatch facilitates bringing them back to these places where sleeping knowledge of clam gardening, interrupted by such impacts of colonization as privatization of land and forced boarding school confinements, is reawakened. Memories and stories come up that wouldn't have otherwise said as elders remember and then share them with new generation, Hatch said. Western science favors distinct disciplines, ecology, biology, geology, and superintendent specialty archaeology, but indigenous knowledge considers the earth, the water, and the air, and the plants, and all the animals as deeply and interdependent and interconnected. So understanding one is to understand all, and that has a lot to teach Western science, Superintendent said, of the importance of braiding these systems. On the flip side, braiding allows Western scientists to use their skill sets to address problems that indigenous communities identify. The fish are unhealthy, or the plants growing in this area are not producing the way we know that they can, she said, and it can provide a second line of evidence to help native nations understand what their ancestral foods once looked like and how to reintroduce them to their communities. Still, the necessity of proving the validity of longstanding indigenous practices can frustrate. Suzanne Greenlaw, citizen Hulton Band of Maliseet Indians, is an ecologist at the Schoodic Institute, a nonprofit of the National Park Service that supports Wabanaki-led research. She participated in a 2016 study that understands how sweetgrass, which grows in salt marshes, rebounds after harvesting. The study was part of a Wabanaki bid to reestablish the right to gather sweetgrass from NPS Island. Though the Wabanaki have made baskets from sweetgrass for centuries, they have been cut off from ancestral marshes in Maine's Acadia National Park for at least 100 years. And that is the end of this week's articles. We hope that they added a little positive joy to your life. Before we end, I have our final segment of the show, something I like to call A Tale of Two Paragraphs. In this segment, I take the first and last paragraph from an article on Substack or another similar source and read them to you, to encourage you to go read the article yourself. Many people forget the power of the written word to move us and how intriguing the middle of the story is. The less I read, the better, but people often want to know how we got to endings more than the endings themselves. And maybe then you will go exploring the Substack and read the paragraphs in between. This week's article is from The Open Letters by Mersalt Substack, entitled I Lied My Way into a MAGA Focus Group, and comes from Patrice Mersalt.substack.com. I like to think of myself as a basically decent person. Responsible, honest. I pay my taxes without trying to outsmart the government. I pay for my own streaming accounts, even when the password field practically begs me to use a family member's login. I rarely drive more than 5 miles over the speed limit. Heck, I return the shopping cart even when no one is watching. So I had a system, a quiet agreement with myself. I'm not the kind of person who does things that require a defense attorney. And yet, this week, I committed an act, premeditated, sustained, and by any reasonable standard, unethical. Furthermore, what am I doing right now? Writing this? Publishing it is, strictly speaking, illegal. A few paragraphs in between, and an ending of, and then came the next question, and with it, something I hadn't accounted for. Up to that point, I'd been operating with a kind of internal script: blend in, establish credibility, wait for the moment to turn. That plan ended there. What followed over the next hour and 45 minutes was not a continuation of what had come before. Conversation moved quickly into places that I hadn't anticipated. It wasn't what I expected. And more to the point, it's not something that I can compress into a few quotes or neat summary at the end of this piece. It needs more room than that. That's part 2 next week. He did it. He did a part 2. Ugh, okay. We hope you enjoyed our show, everyone. Thanks for tuning in. And until next time, I hate to be the bearer of bad news, so I won't. Have a good day.